1 second. All right, so this is going to be recording just so that I can send this out to the instructors. So, excuse me, a couple of these labs are a little bit new, so if I miss anything, let me know. I just got to get your name down. Okay, so my name is Becky. I am the Science Learning Specialist in the Learning Commons. And this lab practical review is for Biology 1020 Intro to Bioanimals, lab practical number 1, which is going to cover labs 1 through 3. So if you have any questions, feel free to stop me, or we can go back at the end. So lab 1 is, excuse me, scientific investigation. So the scientific method is used to test hypotheses to develop a more defined understanding of our biological world. You should know the steps in the scientific method. The hypothesis is your first guess about the solution to your problem based on observations. Oh, I should note that most of this stuff is directly from your lab book. So hopefully that you've read through the introductions. Anything that's bold italicized, you could be questioned on for vocabulary. Again, so hypothesis is your first guess about the solution to your problem, and it's going to be based on your observations. Experiments are developed to test this hypothesis. So you should know the difference between experimental group and control group. The experimental group tests the hypothesis. The control group is in an absence of the thing you are testing. I think in your lab book they used giving treats to a dog to perform certain tasks. So your experimental group would be the group that got the treats. Your control group would be the ones that didn't. So your conclusion is based on data collected from your experiment. After you run through your data and form this conclusion, you're going to either support or reject our hypothesis. And then statistics are used to determine whether differences exist between the control and the experimental group. So again, that would be either to support or reject your hypothesis. So the scientific method enters this cycle where new observations and hypotheses are formed from the conclusions of previous hypotheses and experiments. I apologize, my dogs are home and my dogs are going crazy. So then hypotheses are either disproved or supported. A theory is formulated when many related hypotheses are supported after a lot of scientific testing, and it's a unifying explanation for a broad range of observations. So you have your observations, you form a hypothesis based on those, you develop an experiment to test those hypotheses, you collect data and results, you form conclusions based on that data, and then you revise your experiment to test the same hypothesis or a new hypothesis. So you performed an experiment which correlated between the length of the upper and lower limbs and the overall height of an individual. So you should understand what this experiment was and what you were measuring. So the hypothesis for the experiment is the length of a person's upper limb is equal to 0.4 or 40% of the height of the person. You also should be able to work these formulas that they gave you. So your expected upper limb length would be 40% of your height, or your height times 0.4. And I think that you measured in centimeters. So that might be a question that you get asked: how many centimeters, or what was the unit of measurement for this? And then, so that would be the expected upper limb length, and then the actual percent of height, which is your length of your upper limb divided by height times 100. So you should be able to work those problems using those equations. They might give you a question on this. Again, they might ask you what is the hypothesis, and you would say that the person's upper limb is equal to 40% of the height. They might ask you what unit of measurement you used. So this is an example experiment. I will give you a second to go ahead and read that, and then we're just going to pick out the parts of the science or the pieces of the scientific method. Okay, so a farmer is running an experiment on plant food and tomatoes, and he thinks that he observes that the ones that are given plant food perform better, or have more tomatoes on them. So his observation is that he noticed over his many years of growing tomatoes that plants that receive plant food grow better. So that's his observation. His hypothesis is that plants that receive plant food are going to produce more tomatoes than those that don't. So the control group is going to be the group that does not get the plant food, and then the experimental group is the group that is going to get the plant food. And then, so he's going to run his experiment, calculate his results. And so his conclusion is that plants that receive plant food produce 2 times the amount of tomatoes than those that didn't. So he would, in this case, support his hypothesis. He could revise this a little bit, change the numbers, and run this experiment again to see if he gets the same results and conclusions. This is kind of like a real basic one, but it's just so that you can pull out those parts of the scientific method. And then you guys did your own. So I don't know that you'll get question on your own experiment, but you definitely need to know what you were doing when you were measuring arm length and height, how to find each of those things using those formulas. So some examples of questions that you might get from or on this lab practical would be, what is the relationship between the length of your arm and your height? Again, 40% or 0.4. What is a hypothesis? And identify the parts of the scientific method in the following passage, which we just did a sample one. So that's really the end of lab 1. Do you have any questions on lab 1?
Nope.
Okay, so we're going to go to lab 2. I apologize, this takes a couple seconds. And this is going to talk about the microscope and cells. Okay, so microscope and cells. So you should know the difference between unicellular and multicellular. Cells are the basic units of life. Everything that's living is made up of some sort of cellular form. So unicellular have only one cell in their body. An example of that would be an amoeba. Multicellular are more complex, have more complex body types. They have many different cells that make up the entire organism. So they're organized in a systematic fashion. And these are important to the formation of tissues, organ systems that control the functions of the organism. So you should know the difference between those two. All cells have 3 basic features. So all cells have these, whether they're prokaryotic, eukaryotic, unicellular, or multicellular. They have DNA, which is the hereditary material. They have a plasma or cell membrane, and they have cytoplasm. So some cells obviously are going to have more than that, but those are the 3 basic features that all cells have. So again, you should know the difference between prokaryotic and eukaryotic. Prokaryotic are the simplest of all cells. They lack membrane-bound structures, which are organelles, and those can be the nuclei, the Golgi apparatus, the endoplasmic reticulum, the mitochondria. One of the big features of the prokaryotes is that they lack nuclei. Some examples of those are going to be bacteria and archaea. And then the eukaryotes have distinct membrane-bound organelles. distinct nuclei, which houses the DNA. And these are going to be plant cells and animal cells. So again, we know the difference between unicellular, multicellular, prokaryotic, and eukaryotic, and know the three basic features of all cells. If I run through this too fast, just let me know. All right, so you need to know the organelles in an animal cell, and you also should know their function. Right, the function of the corresponding organelle on the following page. Yep, okay. So hopefully you have this diagram in front of you, because I'm going to skip this so that we can go to the functions of the cell. So animal cells have subcellular structures called organelles, like we talked about, which divide the division of labor within the cell, so the basic necessities of life can be accomplished, such as cellular division, acquisition of energy, elimination of waste, and making molecules for the cell. So the functions of again, hopefully you have that cell picture in front of you. So the nucleus is where the genetic material is. The nuclear envelope, yeah, okay, that is on there. It's a double membrane that encloses the nucleus. And again, these are all membrane-bound organelles, most of them. The nucleolus is where ribosomal RNA is made and assembled with proteins. Chromatin is a complex of DNA that makes up chromosomes. And then the nuclear pore is the little holes in the envelope that regulate traffic within the cytoplasm, or with the cytoplasm. Ribosomes are where protein synthesis happens. Rough ER is endoplasmic reticulum with ribosomes attached, which means proteins are made there because it has ribosomes. Smooth ER lacks ribosomes, and these are the little mazes that you can see in that picture. And the smooth ER is for detoxing the cell and synthesis of lipids. Lysosome is a digestive organelle. Mitochondria is where cellular respiration occurs, and most of the cell's ATP is made. The Golgi apparatus is the sorting and packaging UPS type organelle. It stores and ships products of the endoplasmic reticulum. The plasma membrane is a selective barrier into and out of the cell for the passage of molecules. And then the cytoplasm is kind of everything else, the contents between the plasma membrane and the nucleus and the organelles. Yeah, okay. So if you need me to go back to those, I can. Just let me know. All right, so then you have the compound light microscope, and you need to identify the parts of it and the function of those parts. So we'll start with the base, which is right here, this round circle, and that's obviously what it stands on. The light source is what sends light up into the slide in the objective lenses. The rheostat is right here, and that's what adjusts the intensity of the light. Can you see my little mouse going right here?
Yep.
Okay. The fine adjustment knob is the smaller of the knobs on the side, and that's for fine focusing. And then the coarse adjustment knob is the larger one, and that moves the stage up and down. The stage adjustment knobs are these two right here, and they move the stage forward and backwards and right and left. The iris diaphragm lever is right here, and that adjusts the enhances the optics. The stage clip is what holds your stage in place, right? Or not your stage, your slide in place. The stage is this big square right here, and it's what you put your lens on. The arm is holding the revolving nose pieces and eyepieces to the base. Also, that's when you pick it up, you want to pick it up by the arm and a hand under the base. All right, so the objective lenses, which are right here, Some microscopes have a 4 times and some have a 3.2. It depends on what kind of microscope you have. That's called scanning power. That's the lowest magnification. And then you have a 10 times, which is low power. High power is 40 times, and I don't think any or most of the microscopes that they have in the labs don't have 100 times. So again, scanning power is lowest magnification, low power is 10 times, and high power is 40 times. So when you're looking through low power, you're seeing that that specimen 10 times the size of what it actually is. But when you get into your eyepieces, they also have a 10 times magnification. So if you're on low power, which is 10x, and you have eyepieces, which are 10x, then the total magnification for that objective lens that you are looking at is going to be 100 times magnification. And then the revolving nose piece is where the objective lenses are anchored. Again, so make sure you remember that there's 10x in the eyepieces, and then you multiply that by whatever the magnification is for the objective lens to find the total magnification of your object. I'd be surprised if they don't ask you a question on that. And then we'll just really run through real quick how to focus the microscope. So you want to clean the microscope with a lens paper if it's needed. It's not a bad idea to do it every time. Rotate the lens to scanning power, which either your 3 or your 3.2 times or your 4. Always start and end on scanning power. Turn on the lamp to approximately half intensity. Make sure the stage is lowered all the way down. So that would be your coarse adjustment knob. Turn the iris diaphragm all the way to the right and then partially to the left to open it all the way. Place your slide on inside your stage clip by pulling the metal arm back. Make sure the cover slip of your side is in the center of the stage so that you can see the light going through directly under the objective lenses. Look through the eyepieces while turning the coarse adjustment knob away from you. That's going to move the stage up. It's going to be probably have to be raised almost all the way up, but don't force it. Adjust your eyepieces so you can see what you're looking at. And then once the specimen is in focus using the scanning power, you can use the revolving nose piece to get to 10x or low power. And at this point, you don't turn the coarse adjustment knob. You will only turn the fine adjustment knob to bring that specimen into focus. And then if you're going to examine it under high power, you're going to turn it to 40x. And again, only use the fine fine, not fine adjustment knob to focus. Again, what's the total magnification for scanning power, low power, high power? When you're taking the specimen out, you want to go all the way back to scanning power, lower your stage by using the coarse adjustment knob, and then you can take out your slide. All right, so you were given a picture of some cheek cells and asked to label the cytoplasm, the plasma membrane, and the nucleus. Okay. So one of the questions is this a eukaryotic or prokaryotic cell? If it has a nucleus, you know that it's a eukaryotic. Identify the nuclei. So those are the dark eyeball-shaped structures, and the nucleoli are the dark spots inside those. You can't really tell from this picture too well. I think here is a little bit, this one you can tell the difference between the eyeball-shaped structure and then the darker structure inside. The plasma membrane is what surrounds the cell, and then the cytoplasm is everything else. I always think of it as like a gel holding everything together. Again, this is another question on what's the total magnification. If it was taken with 10 times ocular magnification and 40 times objective, this is going to be a 400 total magnification to look at this. Okay, so that was one of your questions. And because they specifically asked you that question, I would say that this is probably something that they're going to ask you. Definitely, they're probably going to ask you to find at least one of these, or label it. They might point to it and say, what is this? And whether this is prokaryotic or eukaryotic. Okay, so then the next one you looked at was an amoeba, and they really don't have much of a shape. These guys are unicellular. They function as one cell. They don't have a bunch of cells that form organs and other functioning parts. This is just one functioning cell. This is a protist, and it does move. It has a pseudopod, so we could say that this is a pseudopod, and then this kind of engulfs its food. So it's going to pull this in this way and pull it in, but it also uses this as kind of like an anchor. So it might protrude out this way, and then the whole amoeba would move in that direction. Do you see internal structures? Can you name some of them? They didn't ask you specifically what, they didn't ask you specifically to name any. But if you can see internal structures, that means that it's going to be eukaryotic. Is it living? This wasn't on your lab, but if it eats, if it reproduces and maintains homeostasis, those are the 3 characteristics of life, then yes, this would be living. And then again, the question was, if it was ocular magnification of 10 times and objective of 10 times, what's the total magnification? So this would be 100 times magnification. Okay, so be able to identify this as amoeba. Is it unicellular, multicellular, prokaryotic or eukaryotic? And then possibly a magnification question. All right, so then you looked at a rotifer. This is a member of the animal kingdom. It is multicellular. It's a heterotroph, which means it obtains energy from consuming other organisms. This pseudocolum was not a bold italicized word, but it is in your lab. So just to briefly go back over it, it has a false hole. Does this move? Yes, it does move. Does it have internal structures? Yes, you can clearly see internal structures. Is it prokaryotic or eukaryotic? This is eukaryotic. Is it living? Yes, it eats, it reproduces, and maintains homeostasis. Just checking to see. Okay, one of the questions on this definitely is it a living creature? And asks about the properties of life. So your characteristics of life are eats, reproduces, and maintains homeostasis. Okay, so again, unicellular, multicellular, eukaryotic or prokaryotic, characteristics of life. Heterotroph is a bold italicized word, so that's one you should know the definition of. Okay, so the dissecting microscope is used to observe larger specimens. So this one only has a 7 to 45 times magnification. It has more workspace than the compound light microscope, allowing you to dissect without hitting the lens. Allows you to see the surface of objects as well as, large internal structures. See more 3D, and again, useful for viewing larger specimens. So again, you should know where the parts of the microscope are and what they are, what their function is. So your ocular lenses or your eyepieces are right here to see, to look through. Your focusing lens is here. It's only got one focusing knob. Again, that would be to focus the lens. Transmitted light source comes from below the stage. So this is your transmitted light source, and you have a top-down light source right here. So you have 2 light sources coming into that specimen. Magnifying lens adjustment knob is right here at the top, and that's to change what magnification you're looking at. The arm would be the same as the compound light microscope. And then the stage is the same as a compound light microscope. And you have two light source switches because you have this transmitted light source and you have this top-down light source. So again, I think more of knowing what the parts are. I think their function is kind of more self-explanatory than the compound light microscope. Okay, so some examples that you could see from this part of the lab practical. Excuse me, name the microscope part labeled A. It could be the compound or the dissecting. What's the function of that part? Name the cell structure at the tip of the pointer. Again, for the cheek cell, it was nucleus, cytoplasm, plasma membrane. Is it prokaryotic or eukaryotic? Remember, if it has any of those structures, if you can see those internal structures, then it's eukaryotic. How are the compound and light microscope and the dissecting, compound light microscope and the dissecting microscope different? Dissecting is used for larger items. You can dissect under it. And be sure to go over observation questions. So be sure to go over the observation questions at the end of the lab. So one of the questions that I wanted to point out is number 3. How does change in the objective lens aid in your ability to see detail on a slide? So it enhances the optics, obviously. It gives you more magnification, and it controls the light passing through the condenser. Number 6 says list the steps are required to appropriately focus the compound light microscope on an object. I'm not totally sure if they're going to ask you that, or they might give you, here's this, like put these in order of, first to last. But you could see a question on that. I think that's it for lab 2. Do you have any questions on that one?
Nope.
All right, so lab 3 is the last lab, and this is basic genetics. Oh my goodness, what's going on here? Hold on one second. No. Sorry. All right, there we go. Basic genetics. So genetics is a study of heredity, transmission of characteristics, which is transmission of characteristics from parent to offspring. So you should know what DNA is, which is the genetic material in an organism. Genes are where the traits are coded, and alleles are the variation in all of those genes. All right, so Mendel's model of hereditary process. So chromosomes, this is a Drosophila chromosome or a fruit fly chromosome squash at 40 times. So chromosomes are the visible form of DNA when your DNA condenses. Be able to recognize this as a Drosophila chromosome squash. Look for the banding patterns, and you can see all those darker lines filled in by like a little bit of lighter. Each band codes for a particular function, and each band represents one or more genes. Genes are what code for proteins, and what in turn is the coding for your phenotype. All right, genotype and phenotype. You definitely need to know the difference between these. Genotype is the actual variation of the trait or allele. So the actual, what your genes are actually coding, like saying. Phenotype is the physical expression of that genotype, what you are seeing. You definitely should know the difference between homozygous dominant, homozygous recessive, and heterozygous. So homozygous dominant is a genotype with 2 dominant alleles, expressed as 2 capital letters, for example, capital P, capital P. Homozygous recessive is a genotype with 2 recessive alleles, or 2 lowercase letters, lowercase p, lowercase p. And heterozygous is 1 dominant, 1 recessive, so 1 capital, 1 lowercase. An example would be capital P and lowercase p. So heterozygotes, is important to note, usually express the dominant trait. All right, so you guys played a game with alleles. It's a little bit different than the one I'm used to you guys playing. So this time you're taking 2 coins and you're just flipping them, I think a total of 96 times. So each coin, you should know this, each coin represents a parent. You could get asked, what do the coins represent? So each coin represents a parent. Both parents are phenotypically tall and genotypically heterozygous. So, the head side is the dominant allele, which is capital T for tallness, and the tail side is recessive, which is lowercase t for shortness. So both of the parents are going to be tall and heterozygous. So each coin has the genotype capital T, lowercase t. So your parent cross would be 2 heterozygotes or 2 capital T, lowercase t. You definitely need to know that the parents are heterozygous, but phenotypically tall, and what each side of the coin represents. All right, so fertilization is the union of an egg cell with a sperm cell, which results in zygote production. The combination of, so in this case, fertilization is a combination of heads and tails in the zygote, represents the genotype of the potential offspring. So with your cross, with your parents both being heterozygous, the only 3 genetic possibilities are homozygous dominant, 2 capital T's, heterozygous, capital T, lowercase t, homozygous recessive, lowercase t. And you only have 2 phenotypic possibilities, which are tall and short. So remember that heterozygotes express that dominant trait. So the cross, again, between 2 heterozygote parents, your expected phenotypic ratio, what you would see for the offspring, is a 3-to-1, 3 tall offspring to 1 short offspring. And your expected genotypic ratio, wow, I should go back through here and check the spelling on a lot of this. So you have 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. So a 1-to-2-to-1 genotypic ratio and a 3-to-1 phenotypic ratio. And those are the ratios you would get for any cross between 2 heterozygote parents, no matter what trait you're coding for. All right, so analyzing genetic crosses are done with Punnett squares. A Punnett square aids in the analysis of genotypes, which is showing you the principle of segregation and meiosis, where 2 alleles from the gene of each parent separate, so the sex cells only get 1 allele. Normally you have the possible maternal genes on the top and paternal, maternal alleles on the top and paternal alleles on the left. And then your possible offspring go into the cross of the Punnett square. So in this case, this is a pure breeding white flower. White is recessive, so that's lowercase p, p, and purple, a pure breeding purple flower, pea plant. So that's capital P, capital P. Purple in this case is dominant to white. So you would have, you would separate those 2 capital P's in each column. 1 goes in each column, and then the 2 lowercase p's, 1 goes in each row. So if you have 2 pure breeding, a pure breeding dominant and a pure breeding recessive, the only genotypic option for the children or the offspring is heterozygous. And so all of these would be purple flowered pea plants, because purple is dominant to white, and these are all heterozygotes. So be able to determine the genotypic and phenotypic ratios for a cross like this. And again, you just need to look at what you see inside this Punnett square. So the phenotypic ratio, these are all going to be purple. So it's 4 purple to 0 white. And then the genotypic ratio would be 0 dominant to 4 heterozygote to 0 recessive. So then you take 2 of these offspring, so 2 of these heterozygous offspring, and you cross those. So again, we're going to separate those alleles. So if you have two heterozygotes, capital P in the first column, lowercase p in the second column, and then capital P in the first row and lowercase p in the second row. So this is just like the cross with the coins. So you will always get, when you have two heterozygotes, two capital P, lowercase p, you will always get a phenotypic ratio of 3 dominant traits to 1 recessive trait. So these 3 purple are going to come from this capital P, capital P, capital P, lowercase p, capital P, lowercase p, and this 1 white is going to come from this lowercase p, because that is a homozygous recessive. And then your genotypic ratio is strictly just what you see in this Punnett square. 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. And again, these ratios you should know. And if you can't figure them out right away, just do a cross between 2 heterozygotes if you don't have them memorized. So then you looked at some human traits, which were free earlobes, widow's peak, tongue rolling. And for these, you just need to know which one's dominant and which one's recessive. So for free earlobes, the earlobes hang free. Recessive is they're attached directly to the head. Widow's peak is the dominant, and recessive is a continuous hairline. And tongue rolling is also dominant to not being able to roll your tongue. So for these ones, you should just know which one's dominant over the other. All right, so determination of sex in humans. So females are always 2 X chromosomes, males are always an X and a Y. So probability of a man and a woman having a female child. So I was gonna do this cross, but I'm not able to. Actually, I could do this. Let me go in here real quick. Looking for this. Okay, so if we have a male and a female, that is not what I want to do. Just kidding. What is the probability of a male and a female having a female child? Oops. So you have a 50% chance of a male and a 50% chance of a female. Okay. Go back to the PowerPoint. All right, so then they talk about sex-linked traits, and in this one, they are talking about color blindness. So sex chromosomes carry genes that affect the traits other than an individual sex. They are carried on the sex chromosomes, and they're usually inherited on the X chromosome and usually recessive. So color blindness is an X-linked recessive trait. So if you look at this table over on the right, females can have 3 different genotypes. So they can have homozygous dominant, heterozygous, which they call a carrier, and then homozygous recessive. So they have 2 of these can be normal vision, these 2 right here, and then if they have 2 recessive X genes, they will be colorblind. So this is capital is normal, capital lowercase is normal vision, but a carrier of the colorblind gene, and lowercase is colorblind. Males, because the gene is carried on the X chromosome, only have 2 options. If they have a dominant gene, they have normal vision, and if they have a recessive gene, they are colorblind. There's no other options. There's no carriers for the males. Oh, okay. So I'm going to work a couple of these crosses that they did in your lab book just to kind of show you how they work real quick. My pen is not working. Okay. For some reason, this doesn't want to work. All right, let me make a new one here. One sec. Technical malfunction. This doesn't want to work. Well, I guess I'm not going to do this. I apologize. For some reason, my pen is deciding not to work here. I wonder if I can. Huh. Okay, well, I apologize. That's not going to work. So I'll just kind of walk through one of these. We'll go through Joe not being able to roll his tongue. So this is problem 1. So Joe cannot roll his tongue, but both his mother and father can roll their tongue. So remember, if you go back to tongue rolling, so you might be asked to do some crosses with this. So remember which ones are dominant and which ones are not. So for tongue rolling, being able to roll your tongue is dominant. Not being able to roll your tongue is recessive. Joe cannot roll his tongue. So Joe has to be homozygous recessive. The only way you can have the show the recessive phenotype is to be homozygous recessive. So Joe has to be homozygous recessive. If both his mother and father can roll their tongue, then that means they both have to have a carrier of that recessive gene to give Joe one from mom and dad. So parents are going to both be heterozygotes, which means they're both going to be capital R, lowercase r. And I really wish that I could run this cross for you. I'm perturbed here that this isn't working. Let me see if I can get this to work one more time. Nope, it's not going to work. Nope, okay. Well, I apologize for that. So Joe's going to be lowercase r, because he is homozygous recessive, and mom and dad are both going to be capital R, lowercase r, because they have to be able to give, each give him one of those lowercase r's. So what are the chances that a child in this family will be able to roll their tongue? So in that case, you would do a Punnett square between mom and dad, who are both heterozygotes. And then your phenotype would be a 3-to-1, 3 who can roll their tongue and 1 who to 1 who can't roll their tongue. Let me see if there's any. Okay, so they do a couple of sex-linked traits. One, but down on the problem, there's the only member of Arlene's family who is colorblind is her brother. So if her brother is colorblind, then he has to be X with a lowercase b, Y, which means mom would have had to give him that recessive allele. So her father's genotype, because he is the only one that is colorblind, would have to be dominant for that allele, because dad can only give one X to his offspring. So if he has daughters who aren't colorblind, then he would have had to give in a capital B to each of those. So mom's genotype, because he did get it that lowercase b, or that recessive trait from, or the brother got that recessive trait from mom because it's carried on the X, has to be heterozygous. So she's X capital B, X lowercase b. And what is Arlene's genotype if she later has a colorblind son? So if Arlene's not colorblind, but she has a colorblind son, then she is going to be a carrier of that colorblind gene to give to her. And again, I apologize that I can't get my pen working, because that would have made a lot more sense if I could write it out. But hopefully that helped a little bit. So some example questions from this lab: identify the genotype of this individual. That could be any of the genotypes that they went over in lab. Work a cross between a heterozygous plant and a heterozygous plant and show your work. And then remember, that's purple is dominant, white is recessive. Which of the following is a typical phenotypic ratio for a basic genetic cross between two heterozygous individuals? So again, if you work that heterozygous flower plant, you'd be able to see the genotypic and phenotypic ratios. But the phenotypic ratio for a basic heterozygous cross is 3-to-1, 3 with the dominant trait, 1 with the recessive trait. In the allele game, what did the capital T and lowercase t represent? So again, go back to that information. Which of these characteristics is coded by a dominant allele? That could be widow's peak, tongue rolling, colorblindness, and such. Colorblindness is not coded with a dominant allele. What happens to a recessive trait phenotype when in the presence of a dominant trait? Okay, so dominant is always, most of the time, is going to take over. What is the relationship between the phenotype and genotype? Remember, phenotype is what you see, genotype is what is actually coded. What's the purpose of a Punnett square? And which sex is usually most affected by sex-linked diseases? So sex-linked diseases are carried on the X chromosome, but the one usually affected the most would be the male, because the male can only have dominant or recessive. There's no heterozygotes in there. They're not carriers. They can only have one or the other. And again, I was going to work some genetic problems, but my pen is not working, so I apologize for that. Do you have any questions, or is there any of those problems that you want to kind of talk through, since I can't show you how to do them?
I think I'm all good.
Okay, so you're all set? No other questions?
Nope.
Okay, well, thank you. I hope this helped a little bit.